I want to thank Stephen for giving me the opportunity to give some of the reasons for the AI93G growth diet or the AI93M maintenance diet and the arduous process that was involved in arriving at what many consider a critically needed resource for reducing variability and allowing valid comparisons of data from different laboratories and studies using rodent models. I want to start by stating that the late George Briggs of the University of California should be credited with getting the process started that eventually led to the AI93 rodent diets. In 1971, he co-authored a review article with Heather Greenfield that appeared in the Annual Reviews of Biochemistry titled Nutritional Methodology in Metabolic Research with Rats. In this review, they cited many reports that ignored the adequacy and the makeup of experimental diets and the use of purified diets and mineral and vitamin mixes that contain serious omissions or errors in formulas. They then gave examples of how this could lead to variability in results from laboratory or another or questionable findings. In the introduction of this paper, a statement was made that was apparently appropriate today. That is, present-day research techniques have reached such a high degree of sophistication that metabolic events can be investigated at advanced theoretical level. Frequently, however, these techniques are incongruously employed against a background of poor dietary design and inadequate nutritional content with consequent equivocal conclusions. In 1972, George Briggs made the recommendation to the American Institute of Nutrition, now known as the American Society for Nutrition, that the society become a leader in nutritional research methods and purified diets by forming a committee on standards for nutritional studies. The following year, the recommendation was accepted and the first committee was created. In 1974, the committee had its first formal meeting and determined that the aim of the committee was to establish nutritional guidelines for investigators, manufacturers of experimental diets, granting agencies, regulatory agencies, and the others involved in using experimental diets in animal research. At a second meeting held at the National Institute of Health in September of 1974, 8 objectives for the committee were established. I have a letter here from George Briggs listing those 8 objectives if anybody wants to see them. The objectives apropos for this presentation was to produce a document defining diets and related standards and to settle on some actual standard formulas for complete diets, mineral mixes, and vitamin mixtures starting with the rat. The reasons given for this document can be considered still valid today. They include relating the necessity of giving sufficient details in a published article so that the other laboratories and researchers at another time, perhaps years later, could repeat the experiments and thus evaluate and advance the findings reported. The document was also to emphasize the fact that commercial diets, especially those not giving exact composition, containing unrefined animal and plant material, would have variable composition over time that could result in deficiencies or pharmacological effects, markedly affecting the results and conclusions of the experiment. This publication also gave terms and definitions for characterizing laboratory animal diets that I would like to briefly describe here. To start, an open formula is one that gives precise percentage or quantity of each ingredient and must be assembled exactly as specified. A closed formula is one in which the commercial manufacturer does not disclose the exact composition or in type and amount of each ingredient. A proximate analysis does not mean that the combination of ingredients in a closed diet has not changed with market conditions. Cereal-based or unrefined diets or non-purified diets often are closed formulas, but can be open formulas. The 1977 document presented the cereal-based NIH-7 diet, open formula rat and mouse ration as it was called, which is shown here. It should be noted that although the composition of the NIH-7 diet is precisely given, the nutrient composition of the ingredients can vary with the environmental conditions under which the plants were grown and processed, and the variety of specific grains, the source of minerals such as limestone, and the diet, time, and environmental conditions of animals providing the animal-based products. A purified diet is composed consistently of refined proteins, carbohydrates, and fat with added mineral and vitamin mixtures containing approximately pure components in precisely measured amounts. To be useful for replication of experiments, these diets need to have open formulas. The 1977 document described the development of an open formula purified diet known as the AIN-76 diet. The AIN-76 diet shown here was found to meet the known nutritional requirements of rats and mice based on tests of only 6 months. That this diet was found to support growth, reproduction, and lactation comparable to that obtained with the best cereal-based diets. The committee noted that future developments may warrant modification in the diet, and longer testing times would have been desirable, but the demand for a standard diet made it the time to present such a diet. Within a few years after the initiation of the use of the 76 diet, reports of problems with rancidity occurred. The rancidity probably was caused by improper storage or by dietary component additions such as adding an oxidative carcinogen. The rancidity apparently caused vitamin K deficiency. The ad hoc committee recommended that the vitamin K content be increased tenfold in the stabilized form of metadione, that an antioxidant be added to the corn oil in the diet, and that the 50% sucrose component be substituted or partially replaced by glucose or starch, especially for long-term studies. With these modifications, the diet was renamed the AIN-76A diet. For 10 years, the AIN-76A diet was a well-accepted guideline for nutritional experiments with rats and mice and helped standardizing studies between laboratories. However, once again, some nutritional and technical concerns appeared about the AIN-76A diet. And a major concern was that there was no definitive recommendation for the change in the sucrose content of the diet. As a result, a 50% sucrose diet was often used in long-term studies, which resulted in increased blood lipids, especially triglycerides, and caused fatty livers, and increased oxidative stress, such as might potentiate the need for some other nutrients such as copper and magnesium, and cause glucose intolerance. Another concern for long-term studies was the diet caused kidney calcification, particularly in female rats. The nephrocalcinosis affecting kidney function could lead to pathology such as hypertension. This is when I came into the picture. I was asked by the American Institute of Nutrition to chair a new laboratory animal diet committee in 1988. The charge to the committee was to determine if enough new knowledge had come forth to make it time to reformulate the AIN-76A diet. The committee decided it was time and asked me to lead the effort. I believe it was at this meeting that it was suggested that a workshop be held in the 1989 American Institute of Nutrition annual meeting. At the May 1988 meeting, after the May 1988 meeting, I began contacting individuals to participate in the workshop that I would chair in New Orleans in 1989. It was then I realized that the reformulation of the AIN 76 diet really needed a commitment of time that me as a director of the Grand Forks Human Nutrition Research Center, I did not have. It is then I made one of the smartest moves in the reformulation process in that I asked the scientist at my center, Dr. Philip Reeves, to help with the workshop and with a follow-up effort that was likely to ensue. Now, to start the discussion at the workshop, 10 committees of 3 with expertise in the Erexine diet component presented recommendations for changes in the reformulation. The 10 components were amino acids and proteins, carbohydrates, fats, fiber, fat-soluble vitamins, water-soluble vitamins, macrominerals such as calcium, phosphorus, magnesium, sodium, potassium, chloride, and sulfur, trace minerals such as zinc, copper, iron, manganese, and iodine, and ultra-trace minerals, chromium, fluoride, selenium, boron, nickel, vanadium, and silicon. And then there was a miscellaneous group which was responsible for such things as mixing, storage, the other additives, and education. The formal presentations were followed by informal discussions by each of the groups with contributions by the other members, interested attendees at the workshop. The outcome of the workshop, which we gave the major reasons and suggestions for reformulation of the AIN 76A diet, was published in the Journal Nutrition later in 1989. This publication stimulated numerous telephone calls and letters about the reformulation. Discussions at the workshop and comments received by Phil Reeves and myself were critically evaluated, and Phil Reeves essentially came up with a new formulation for the AIN diet. Now, after we had the reformulation together, a request from me and Phil Reeves that funds and laboratory be found to validate the adequacy of the new formulation was sent to the American Institute of Nutrition. The request was not supported. In 1990, I made a decision as director of the Grand Forks Human Nutrition Research Center that having a diet that supported the conception, gestation, lactation, growth, and expected lifespan without noticeable pathology was needed to perform experiments that gave valid findings that could be used to design experiments determining the metabolism, function, and requirements of trace elements in humans. Thus, I informed Phil Reeves that the center resources could be used to evaluate the new diet formulation and be considered a component of his research project. Phil's initial formulation, which was first called the AIN 89 diet, and then the later AIN developmental formulation or AIN DF diet, still had some problems with kidney calcifications, concern about arsenic, which was considered a possible ultra-trace mineral at the time, accumulating in the erythrocytes of rats, and questions about substituting L-cysteine or L-cysteine for L-methionine to meet the recommendations for the sulfur amino acids. This is the final composition of the AIN 93G diet, and is shown alongside the AIN 76A diet. Notice that L-cysteine was used. Also notice the differences in sucrose, 100 versus 500 grams. Cornstarch was changed from 150 to 397.5 grams per kilogram, with an additional 132 grams of dextrinized cornstarch. The fat source was changed to soybean oil and increased from 50 to 70 grams per kilogram. Changing the molar ratio of calcium to phosphorus from 0.70 to 1.30 eliminated the kidney calcification problem. This was done by substituting calcium carbonate for calcium phosphate. No arsenic was added to the mineral mix, so this eliminated the arsenic question. A final issue was to get the formulation into a publication so that it could be used. The request to publish the development and testing and the composition of the newly formulated AIN 92 diet, as we call it, and technical aspects in preparing and modifying the diet as an article in the Journal of Nutrition was forwarded to the American Institute of Nutrition Council. The council came back with the statement that it had concerns related to about associating the name of the American Institute of Nutrition with a diet formulation. And secondly, the council also indicated that it had concerns about the final formulation and testing only arising from the Grand Forks Human Nutrition Research Center. However, the decision about publishing the article became a pressing issue for the AIN Council when Phil submitted a manuscript to the Journal of Nutrition reporting his findings using the new formulation. The AIN Council then decided that the formulation should be a report of a writing committee that, in addition to Phil Reeves and me, had an AIN member that would review the validity of the report that we had written and the development and testing of the formulation and the appropriateness of the recommendations made in the report. This member was George Faley at the University of Illinois. The AIN Council also indicated they wanted the report clearly indicated as a report and published simultaneously with Phil's article on testing the diet in a 1993 issue of the Journal of Nutrition because many AIN members and the others are anxiously awaiting the publication of the AIN 93 rodent diet. So there you have a sense of the arduous process that Phil and I went through to get the formulation in place. Also note, even with continued pushing of the issue by Phil and me, it took 5 years from the initial decision that the formulation should be updated until the final formulation was published. Publishment was done before long-term feeding and reproduction testing was performed, which was originally considered a requirement before the diet could be a standard for studies with the nutritional component. However, maybe I should say luckily, the use of the AIN 93 diets without any significant issues the past 25 years indicates that the decision to get the formulas published was right. The AIN 93 diets addressed most of the features discussed in the workshop and the commenting period during its development. The diet had unsurpassed reproducibility that could be prepared with ingredients easily available and at a reasonable cost. It could be pelleted to prevent animal wastage and was stable with a long shelf life. It was palatable and precisely provided all known essential nutrients in required amounts. It could be easily manipulated to study the impact of individual nutrients and allowed the addition of metabolism modifiers, anti-nutrients, and possible toxins in a controlled manner. The report also gave some precautions about making manipulations in the diet and important factors in the preparation of purified diets. Now, the basis for the AIN 93 rodent diets is still relevant today, and is the basis for the examination, and just a cursory examination of the recent literature reveals many ways a difference in the composition of diets can affect experimental results. For example, the amounts of proanthocyanins or oligomeric flavonoids which can affect reactive oxygen and lipid metabolism response in a study varies with the types and species of cereals used in a cereal-based diet. In addition, cereal grains-based diets can have different quantities and types of 18-carbon fatty acids that can alter oxidative and thermogenic responses. How a plant component is processed can also markedly affect such things as energy and amino acid or protein digestibility. Recently, it was reported that 3 different chow diets differently affected the composition of microbiota in their mouse gut, which could affect their metabolism. These examples show that a difference in diets fed to animals for which a variable is being examined can be affect the validity or the repeatability as to the cause of any change observed or not seeing any change. The need for the standard diets is generally well recognized. The AI93 report has received almost 6,000 citations, and I've received 2 to 3 notifications each week that it has been cited. However, according to a recent emission of BMC research notes, it was stated that 70% of researchers cannot reproduce another scientific experiment. Well, some may question this high percentage, but it does indicate that there still are problems in duplicating research findings. There are several possible reasons for this, but I suspect that the lack of attention to the nutritional aspects of an experiment probably is the cause for some of the difficulties in the repeating of research findings. Now, I'll give you an example of an improperly controlled experiment is when a chow or cereal-based diet is used as a control for comparison with animals fed an open purified diet, or just stating that a chow diet without a description of its content. In the next few slides, I will use the open NIH7 natural ingredient diet because it gives an idea as to the amount of nutrients that could be in a cereal-based diet. I will compare the nutrient composition to that in the open purified AI93G diet. This slide gives the average macronutrient composition of the NIH7 diet. Now, this is not much different than the AI93G diet, which contains about 20% protein, 51% carbohydrates, 7% fat, and 5% fiber. Now, this is where the similarity ends. The amino acids required by animals is supplied by a relatively pure form, casein, in the AING3 diet. The amino acids come from skim milk, fish meal, soybean meal, alfalfa meal, corn, wheat, and torla yeast in the NIH7 diet, and the amount of each amino acid is quite different than that in the AI93 diet, and this difference can vary with the source of these numerous ingredients. Moreover, some of the protein sources contain biologically active phytochemicals such as isoflavones and significant but varied amounts of essential minerals and vitamins. The fat, which comes from different sources, provides less linoleic acid and omega-3 fatty acid with an N6 to an N3 ratio of 31.4. The AING diet provides 3.57% and 0.48% respectively, with a ratio of 7.4. The desirable ratio for rodents is less than 10. Notice the high amounts of this slide shows the mineral composition of the NIH7 diet. Notice the high amounts of certain minerals such as iron, manganese, zinc, and copper. Perhaps the high iron content is appropriate because manganese interferes with iron metabolism. Perhaps a high copper is needed because zinc apparently interferes with copper metabolism. In any case, some of these minerals appear to be provided in supernutritional or pharmacological amounts. This slide shows the amount of minerals provided by the AING diets. Note that all are provided near the recommended or required amounts, and these amounts are consistently provided. These include calcium, phosphorus, potassium, magnesium, and the trace elements. It should be noted that some mineral elements that have been found to have beneficial or favorable effects on trace amounts for rodents were added to the diet to assure that amounts of these minerals did not vary greatly in the AI93 formulation. In retrospect, recent research indicates that only silicon, boron, and fluoride probably should have been of concern. The amount of these elements are likely to vary in the NIH7 diet and can be low or high in any natural ingredient diet. Interesting, you got to realize that fluoride is in some tap water, and that is often used in some experiments, and the other experiments that use purified water, which does not contain fluoride. It also should be noted that boron has been reported to be essential for two animal species, the zebrafish and frog. Differences in embryogenesis occurred in zebrafish fed a natural ingredient, boron-deprived brine shrimp, and maintained for 6 months in water containing only 1 micromoles of boron per liter, when compared to those fed the same shrimp and maintained in water containing about 45 micromoles of boron per liter. The early cleavage stage of development was most sensitive to boron deficiency. 46% of the fertilized boron deficient embryos did not complete the blastocyst stage, compared to only 2% for the boron adequate embryos. During this time of rapid division, cell division, the boron deficient zygotes exhibited blebbing of the cell membranes, followed by cytoplasmic and yolk extrusion. It also should be noted that adult F1 boron deficient zebrafish exhibited photophobia. Now this slide shows the average vitamin content of the NIH-7 diet. Note the high amounts of safe vitamin D. The AING diet, it's a 93G diet, provided the vitamins near or the required or recommended amounts. I would like to summarize my presentations about the AI93 diets and perhaps follow with a few additional comments and suggestions. The formulation of the AI93 diets was a culmination of 20 years of efforts of many members of the American Institute of Nutrition, now known as the American Society for Nutrition. The diets have become a paramount resource for reducing variability and allowing comparisons and data from different laboratories and studies using rodent models. One has to believe that the AING diets have abated the appearance of unjustified conclusions and unrepeatable experiments in the scientific literature. It is still relevant 25 years after its appearance, and thus could be an example of an importance of such a diet for experimental models other than rodents. I just would like to make a couple comments. If a natural ingredient has to be used in diets for aquatic models, for biomedical research, attempts should be made to make the most as consistent as possible. And a report about a standard diet should be caution users that the environment may alter the response to the diet. And this could be a major issue for zebrafish because zebrafish lives in water instead of air. And a report giving a standard diet for animal model also should make the readers who are inexperienced in such matters aware of some precautions in the use of the diets. If substitutions are being made to the diet, such as macronutrient content, for example, to make it obesogenic, the method doing so requires some thought. When the alteration in calories is derived from fat or alterations in fatty acid composition, it should be made on an caloric basis and not by dilution or substitution. The report should give guidance about diet preparation if it is not obtained from a commercial source. The goal should be to prepare a diet that is homogeneous, especially in regard to those constituents present in extremely low amounts. And mixing should result in a minimal loss of labile ingredients. Finally, the proper storage of diet should be indicated in the report. I would recommend that diets containing defined purified substances be stored refrigerated in covered plastic containers. The storage probably should not exceed 3 months. The diets could be stored frozen to prevent deterioration, but this probably should not exceed 6 months. I would like to end by stating that it is becoming increasingly aware that there are complex interactions among nutrients, environmental conditions, and physiological states. Thus, the needs for a defined diet is paramount for studies of metabolism with a nutritional component. Thank you very much. Any questions?
The floor is open for discussion.
Yes.
Was there ever any consideration for the optimal diets at different ages or life stages or the different sexes?
Well, yes and no. As you notice, I said we had an AIN93G diet, and that's for growth. And there's also an AIN93M diet, and that's for maintenance. That's for keeping animals maintained after they have grown and become adults type of thing. We have found that there was essentially no difference. We've got both males and females, and there was no difference in response to how they compared to those that are well-based, cereal-based diet. So sex and different stages were somewhat considered in the formulation. Yes.
Can you comment on how the commercial providers dealt with this? Like
What's that?
The commercial providers, like the actual chow makers, how did they interact with your guidelines, and how did they all just start making it available, or was there a challenge there?
There was one group, one new maker who really supported this well. I got the name of it in my folder there. I think it was Research Diets, and they said they would make it available to people who wanted to use it, and it became the demand for it came quite great from this one commercial user, and it eventually led to the other commercial companies producing it for the investigators. I can look up the name of the company that really start getting that into the researchers' hands.
But was there existing products that were already out there that were commercial products that, in a way, you had?
So I can't hear you.
Were there additional products already out there that people were using? They would buy,
Well, yeah, they were using the AIN76A diet.
and that was being sold.
And then they also, a lot of people who didn't realize that standard diets are useful for reproducing your experiments were buying good old rat chow diet from Purina, made by Purina, for example, and then using that as to do their experiments, and sometimes comparing that to an AIN76 diet, which is ridiculous. But yeah, they had chow diets, and this, when the AIN76 diet first came out, there were still a number of mineral mixes, vitamin mixes, and various type of diets named after various investigators that developed them available from the commercial companies. But these went by the wayside as soon as the AIN76A diet became well accepted.
You mentioned that it took about 20 years from initial advocacy.
It's when George Briggs suggested that they should do such a thing, development of the AIN76, then the 76A, and then the 5, it took about 4 years. It was used about 10 years to find that there were some errors and some problems. And then the AIN93 diets came in 5 years. So it took 20 years before we come up with a formulation that's been used for 25 years.
My question is, then, is there any information or guidance that you could provide us in terms of reducing that amount of time?
Well,
expediting things somehow.
like I say, I think it's going to take a commitment of 1 or 2 individuals, really, like Phil Rees and myself, and for the AIN76 diets with George Briggs and John Bury. It took 2 people who really put a lot of commitment in pushing to get the things going forward. And that's what's really required. But they do need the input from the community that's interested in the standard diets that's being formulated. So getting comments and suggestions from that community is very, very important. Do this either by a workshop like we did, inviting in people who had expertise in various nutrients for the fish, or to make sure that and today you can get it by email. For us, we got it by letters and telephone. They didn't have email 30 years ago, let's put it that way. So I have a lot of letters along here if anybody wants to see what kind of comments we receive. Yes.
Did you, when you established this diet, did you come up with recommendations as to how often they should test every batch or every lot? Was there recommendations involved with that, or did that just progress naturally?
We never did that. But, about the only recommendation we made, that if you're going to store the diet for some time, you should test for oxidation deterioration of fats, and if there is some rancidity or oxidation of fats, it should cause a breakdown of vitamins. So this should be tested for. We reported that in the AN 76 AN 93 document, that this should be tested for occasionally if it's held for a long time. Yes.
So you mentioned reproducibility.
Yeah.
For reproducibility, in the mouse model, we deal mainly with isogenic strains. In the zebrafish world,
I'm not here as well as I used to.
Sorry.
Yeah.
So in the zebrafish world, we hardly ever deal with isogenic strains. We have very diverse fish strains.
Uh-huh.
In terms of reproducibility, in the mouse model, they are very isogenic strains, and does the diet have an influence on reproducibility more than, for example, in zebrafish?
It could be, because there could be different requirements for some of this different genetic strains of mice. So that's what you're saying.
Yeah.
But it's still, the basic starting point is a standard diet, so that what, if they're not doing well, then there's something probably that needs to be changed in terms of a mineral or a vitamin or something like this, or amino acid. Like that. At least with a purified diet, standard diet, you can look at each ingredient and see which one is not working for that mouse, or for example, for your fish.
I just want to say, I just want to bring up an issue I have, and that we're throwing the term chow around here, and actually chow is a copyright term for purina diet.
But they use it in the literature, and
But
they're not dealing with
It
purina
shouldn't be used in the literature, it should be diet or feed, but chow is that's just, something that I was taught by Joe Kanapka, who was, one of the premier,
Which diet group are you with? Are you with a diet group?
no, with NIH.
Okay,
So I just wanted to say that chow is, Joe Kanapka told me never put chow in the paper.
It's a commercial.
And Joe Napa was the one that developed the NIH 7 diet, by the way.
And the 31, and there's 13 other open formula diets.
So on that point, it sounds as if there are still in the field a number of different open defined diets that are being used by different groups in the rodent field. Is that true? And if so, has that been?
Well, the AIN 76 diet, the A diet's still available if you want it, and AN 93 diet's available. They still sell closed formulas, and I think they have some open formulas which are made from soy-based and animal-based products. I haven't looked at that very closely.
But, well, I guess it gets back to, do people use these other formulations, and do the papers get rejected or something? Like, is it a requirement to get, like, when it goes out for review, if they're still all out there?
If they use the AIN 76 diet, they're not going to reject it. It's a defined diet.
Right.
But the people who read that realize that there are some concerns that could be involved with, in terms of kidney calcification if it's, for example, used in a long-term study. Short-term studies probably would be all right. But what happens so often in papers that I reject as an editor of a journal is they'll say, we've got a chow diet. That's it. You have no idea what was in the diet, or they've got to purify it. If you're going to have some publication done, the diet should be defined, so the next person along the line can repeat. That's what the same kind of components in the diet.
I'm just trying to get an idea how widely accepted in the rodent community are, like 80% of in every university, are they using an open diet, or they're still
Best
using?
Best thing I can do for you is, like I tell you that, I don't know if they even got all the citation zones, but since it come out, over 6,000 sites have come for the AN 93 diets, which makes me a highly cited author, by the way. And I get 2 or 3 every week, and generally, people who know that a standard diet is going to be used are using the AN93 diet. For the uneducated in the use of nutritional parts of the experiment, there's a lot of people out there that we get articles from that we say, no, this is not acceptable. We have to know what is the nutritional component and the standard conditions are for the animal so that it can be repeated by other people. Yes.
And I think at the same time that they were coming up with the AIN76 diet, that the American Institute of Nutrition suggested that as a standard reference diet, the NIH 07 and I think the NIH 31, which is the autoclavable rodent diet, they should be used as a natural ingredient standard reference diets at that time.
I'm not sure about
I am. I wrote a paper. It's published.
I guess I have a question for the NIH folks, just to Steve's point, is there ever going to be a top-down edict that, these standard diets should be used or else, right? Your grant won't be funded or your paper won't be published?
I think it really should. The nutritional component is necessary if you're going to be able to reproduce the results that are getting from the research in that grant or in the paper.
I'll respond to that a little bit. Cindy Davis on the ODS, and I should thank you, Crossley, for a very nice presentation. I think there is a big push to increase nutritional reporting and a realization that will help to it. Part of the problem is nutrition is still such a small component, and not everybody realizes how important it is. And even as a nutritionist, and really appreciating the need for purified diets, particularly stuff like the AIN93 diet. For any microbiota research, the AIN93 diet is not appropriate. It uses cellulose as a fiber source, which isn't metabolized by gut microbiota. In rodents, it's not the appropriate fiber source. So there are tweaks, there are reasons not to use it. I think the biggest key that Frosty said that we really need to think about is anytime you can standardize things, it improves things, and you really need to know what is being used, and so that people can reproduce what you're doing.
And like I say, the AN93 diet, you know what's in it, so you can change things to make things what you want to happen in the experiment.
I just want to say that up until the early 2000s at NIH, you had to, you were expected to use one of the NIH open formula diets in your research unless you could come up with a scientific justification for using a closed formula diet, something off the shelf from one of the feed manufacturers. However, that has changed, and there are people who don't understand the significance of it or advantages of an open formula diet, and not everybody is feeding the NIH open formula diets, and you don't have to write justifications any longer. But there was, when Joe Kanaka was around, that was the way it was. So.
Yes.
Is a completely chemically defined diet, has that ever been developed for rodents?
Chemically defined diets have been developed. That's when you use purified amino acids essentially in addition to, instead of casein. And that's essentially all it is. And
But that's not.
I just don't know where that would be published. I've been involved mostly with the standard purified diet that you saw here. We mentioned the purified diets in our report. I'll have to go and look at it again.
But that's not true. It's not just
the chemical defined diet.
It's just not the protein, but there's the soybean oil. There are a number of ingredients in there that are complex ingredients still.
Yeah.
So has any chemically defined diet or hylitic
Yeah.
medium ever been completely successful at recapitulating?
I don't think there's anything that's successful, but there's some been made, and it does support growth, but it does not do as well as your standard purified diet or your open formula natural diet.
There's an issue with palatability at that point, isn't there, when it's all chemical?
Yeah, that's part of it. Yeah, cost too.
As well.
Forrest, thank you very much, and that this discussion is going to continue. We can tell by the discussion already that it's very valuable to us.